Hello, everyone. First of all, I apologize for not being able to be there. I would have loved to be there with you and visit Montenegro. Unfortunately, the timing didn't work out very well, and I have to record this talk online. Uh, Apologies in advance. I'm going to talk about accelerating genome analysis in 10 to 15 minutes. This is an area that's very close to my heart, and this is an area where we're doing a lot of research on. I see this as an ongoing journey, and it's a very important problem for humanity, in my opinion. We're going to talk about system design for bioinformatics a little bit. I believe this is a critical problem and has large scientific, medical, societal, and personal implications. We're going to focus especially on genome sequence analysis, and in particular, read mapping, although we do a lot of other work on genome analysis in general. And I will hopefully show you that many bottlenecks exist in accessing and manipulating huge amounts of genomic data during analysis. And we will cover some recent ideas in accelerating read mapping, and I will provide you with my journey since September 2006. I believe there's a lot more to come in this direction. So what happened in 2006? Basically, we had imagined, we were dreaming of an embedded device that could perform comprehensive genome analysis in real time, hopefully within a minute, answering complex questions like this: which of these DNAs does this DNA segment match with? What is the likely genetic disposition of this patient to this drug? What disease or condition might this particular DNA or RNA piece may be associated with? And many, many other things that could potentially be life critical. And to answer these questions, we really need faster and scalable genome analyses. And there are many, many questions like this, as you can see. And as COVID-19 pandemic has proven again, disease outbreaks are an example of it. Personalized medicine is another example. Microbiome understanding, disease understanding, and many, many other applications are examples. And also understanding evolution and species. I believe there's a bright future for this because today we have these devices that are very capable of generating genomic data, but we need to make them intelligent and enable them to actually process the data also efficiently and scalably. So for this, we need across the stack hardware, software, co-design, algorithm, architecture, device co-design going into the future. And that's going to be the theme of my talk. I'm not going to go into a lot of detail in many of the ideas. I will refer you to some papers, overview papers that we have written. This is one example. This is another example. And this is a more recent example that was invited for the special issue of this journal. If you're interested, you can take a look. But I'll try to give you a brief overview, hopefully relatively quickly. I have a lot of slides, but I will try to give the gist of the ideas. So basically, clearly, we're dealing with genomes that make up a lot of our traits, as well as deficiencies and also vulnerabilities. And these are real, clearly, they exist, and they exist in many, many beings. And specifically, we're interested in particular portions that actually lead to diseases, vulnerabilities, reactions with drugs. And we would like to really identify and understand these portions. And this is clearly a big scientific and medical problem. And this genome exists in many, many living beings, all living beings, and they have different properties. And understanding those living beings is also very important. So I'm going to focus on DNA, but this applies to RNA as well. The goal of DNA sequencing is to find the complete sequence of bases or base pairs, A, C, G, Ts in an organism's DNA. The challenge is, unfortunately, there is no technology that we have developed so far that takes DNA as long input and gives the complete sequence as output. If we had the complete sequence as output, we could query it much more efficiently. Unfortunately, all sequencing machines are choppers. They chop DNA into pieces and identify relatively small pieces, but not how they fit together. So we have a computational puzzle when we're answering all of these complicated questions that build on understanding the DNA. So genome sequencing essentially leads to many small DNA fragments, which we really need to identify. These are called reads, and map to a reference genome, for example, or assemble in some other way. And then ask the questions: what is this part of the genome like, and how does it react to a drug, for example, that we know of particular interactions with? So I've already said this basically. I liken this to untangling yarn balls. You have these yarn balls that are different DNAs, and then you basically sample them and you try to figure out what is the organism, for example, or what kind of yarn ball you're dealing with, and what kind of disease a particular organism is vulnerable to. And these are the examples of genome sequencers today. There are many, many genome sequencers. There are small ones, there are big ones, there are expensive ones, there are cheap ones, and they have very different properties. And these have enabled, the technological advances in genome sequencing have enabled a genomic data explosion. So basically, with the introduction of high throughput sequencing technologies, the cost of genome sequencing has reduced greatly. And more recently, with the introduction of nanopore sequencing technology, that has also reduced even greatly. So you can see that the cost of a genome is reducing much faster than the cost of a transistor. So if you'd like to read these, you can find more data from going until 2022 today, but I'm not going to talk about that. So, but basically, we have a puzzle. We actually want to construct the entire genome, but we have a puzzle, so we need to really complete the puzzle. And this puzzle tends to be a computational problem. And newer genome sequencing technologies actually complicate the problem because they have long reads, long pieces, but unfortunately, they have high error rates as well. So there are many different sequencing technologies. I will not go into a lot of detail. Technology is very, very fascinating, but in the end, technology is not perfect. So we need to make up for the imperfections of the technology with our computational techniques across the hardware and the software stack. I'll mention some of these computational techniques. So essentially, today we have very good sequencing machines. They're not perfect, but we need to figure out what they're producing. And that's a computational task. And on top of that, after that, we need other computational tasks to understand what the genome actually means, which will lead to scientific discovery and medical advancements. And the first step in genome sequencing is really read mapping, understanding what his genome looks like. And there have been many algorithms that have been developed over the course of 40 years. And we covered this. We cover actually a lot of these algorithms in the survey paper. If you're interested, you can take a look at it. I'm going to focus on one problem now, which is the read mapping problem. Sequencing machines gives data as chopped DNA. How do we actually reconstruct the DNA? And today's machines are bottlenecked in this read mapping problem. As you can see, there are some numbers over here. Today's machines are actually quite bad at this because genome sequencers are actually very special purpose machines. They generate lots of data very quickly. Unfortunately, we process that data using general purpose machines. As a result, we have a mismatch. We have very slow and inefficient processing capability that doesn't match the throughput as well as the latency of production of genomic data. So how do we actually fix that problem? Basically, we need to reconstruct the entire genome from many sequence reads. And this basically looks like this. You have physical DNA like this. You chop it into pieces, you get reads, and then you need to map these to a reference genome or somehow reconstruct a reference genome through assembly techniques. I'm not going to go into a lot of detail, but there's a lot of work, algorithmic work. And this becomes even more complicated for metagenomic analysis. These are the iron bores examples that I showed you. You may actually recover genetic material from environmental samples, from a subway, for example, when you're doing outbreak prediction. And then you actually have reads in text format, and then you need to map them to a reference database of many, many genomes and identify different pieces, which pieces belong to which genomes, essentially. This way, you can identify potential outbreaks, potential viruses that are lurking around in a subway, for example. This is called microbiome sampling. And this is this computational cost of edit distance computation, alignment, in other words, is mathematically proven. Basically, it's tough. And that's why people have developed a lot of algorithms. And today's genome sequencing pipelines actually spend a lot of their time in read mapping. I will not go into detail. You can read a lot of these papers. So we've been focusing on accelerating this particular part of the genome sequencing pipeline so that we can accelerate the entire pipeline. And there are many portions of it. I'm going to give you some ideas of this, but there's detailed analysis in some of these overview papers. Now let's talk about the ideas. So certainly you can accelerate things in algorithms. I'm not going to talk about that. You can exploit the structure of the genome. You can exploit SIMD instructions, etc. I'm going to focus more on the hardware acceleration, but hardware acceleration also tends to be hardware-software co-design. In the end, we're going to talk about specialized architectures and more about the future memory-centric computing, like processing in memory and storage, and then briefly talk about future opportunities and then conclude. So we have been looking at a lot of FPGA, field programmable gate array-based alignment filtering and alignment opportunities. And this is one paper that I will briefly mention. Essentially, this is the first FPGA-based alignment filter. The goal is to actually minimize the useless computation that you do in the genomic analysis pipeline by quickly filtering things that will not match the reference genome. And that's the idea. We have some algorithms that are implemented in FPGA. As you can see, our real results are quite good. End to end, we can get an order of magnitude speed up in read mapping. And these are all freely available online, so people can actually build on it. So I'm going to skip this one, but basically, this can enable on-device genome analysis by doing things much faster and also energy efficiently because it can enable real-time filtering while sequencing. So clearly, it's an example of an algorithm architecture device co-design, and everything I will describe is actually will be an example of that. We need to really rethink how we do the genome analysis all the way from algorithms to devices and co-design across the transformation hierarchy. Later, we developed a lot of other algorithms. This is another example of an FPGA design that I will not go into, but this exploits some insights that are actually, you can build a sliding window-based alignment algorithm in FPGA. And this turns out to be actually much faster than prior algorithms. And more recently, we also looked at another algorithm that actually transforms the problem, approximate string matching problem, to the single net routing problem in VLSI chip layout. And as a result, accelerates the problem significantly, more than an order of magnitude, another order of magnitude, essentially. I'm going through these relatively quickly, but this hopefully gives you an idea of the algorithm architecture co-design characteristics. I will briefly mention this one. This is the first approximate string matching acceleration framework that can accelerate multiple steps of the genome sequence analysis pipeline. And this is based on an algorithm that was developed 30 years ago, by TAP algorithm. This uses fast and simple bitwise operations to perform approximate string matching so that you can compare different parts of the genome. And we modified and extend this algorithm so that we can make it highly parallel. And also we enable traceback so that we can actually do edit distance computation on this. And we co-designed this with memory efficient algorithms with area efficient hardware accelerators. I will not go into the details of this, but this is actually a sophisticated hardware design that has relatively low overhead, as I will show you. This work was done together with the Intel Corporation, and they're quite interested in actually building some of these things. And you can see that the area and power overheads are relatively low. And this actually accelerates multiple steps of the genome analysis pipeline, including edit distance computation, which is not exactly shown here. But you can see that the speedups are actually quite high compared to existing software that's commonly used. They're also quite high compared to existing hardware while having less power consumption. And you can see that other use cases also see significant speedups compared to existing software and hardware algorithms. You can read more in our paper clearly. More recently, we've been looking at graph-based genomes, and this is actually the future of genomics because you represent genomes using graph structures, because people are interested in actually representing a population using graphs. And this turns out that this complicates the problem significantly. It's a much more difficult computational problem because now you don't map to a single reference, linear reference, you really map to a graph. And we designed the first algorithm hardware co-design that alleviates the problems, bottlenecks in multiple steps of the sequence to graph mapping. And this is actually a universal algorithm hardware co-design that can actually do both sequence to graph mapping and sequence to sequence mapping for both short and long reads. And you can see that the graph mapping, sequence to graph mapping pipeline is more complicated. You deal with graphs in every stage of this pipeline. And again, I'll give you some key results. This is a hardware accelerator, specialized hardware accelerators, algorithm hardware co-design that leads to significant speedups and significant power and energy savings compared to existing tools, as well as existing accelerators for multiple different use cases, as you can see over here. And you can find out more on this topic. My student Damla did her PhD on this topic, and you can find out her ISCA talk. Now I will quickly jump into processing and memory storage, which is more future, like memory-centric computing. Basically, this is a very memory-bound problem. As a result, we need to rethink the problem such that how can we, to think about minimizing data movement. How do we minimize data movement, and how do we do in-place computation, in-sensor computation, as well as in-memory computation? To be able to do that, we need to design mapping and filtering algorithms that fit processing in memory or processing in storage. And again, we've done this on real systems using FPGAs that have high bandwidth memory access. And we've shown significant speedups compared to very powerful, hefty systems like IBM Power 9. This is a paper that describes both performance and energy efficiency improvements using real systems, real FPGAs, when we implement Sneaky Snake algorithm that I mentioned earlier on FPGAs. You can read more about that. I will briefly talk about storage because as the genomic data explodes and explodes and explodes, it doesn't fit in memory structures. You need to really fit in storage systems. And storage systems actually today is a huge bottleneck because when you need to move the data to a computation unit, you have to go through the storage system all the way to main memory caches and computations. So computation overheads and data overheads are both exacerbated. Now we try to alleviate computation overheads today using many, many algorithms that I just described, heuristics, accelerators, and filters. Unfortunately, data overhead, data moment overhead from the storage system remains. The key idea in many of these, in this work, as well as going forward, is to filter reads, filter some sequences that do not require alignment inside the storage system. Basically, do some of the easy filtering in the storage system so that the bottlenecks are alleviated in other parts of the system. That's the key idea. And then there are details on what should be filtered. You need to be careful about what to filter so that you don't lose information. And we talk about that in the paper in more detail. You will need to take advantage of the sequencing machines as well as the input data set, which I don't have time to go into right now, but you can read the paper. So the challenge is basically taking advantage of the workload behavior as well as limited hardware resources in the storage system. And in the end, we show that you can actually improve significant performance, lead to significant performance improvements and energy reductions by filtering reads inside the storage system so that other parts of the system is alleviated. And this is actually improving both performance and energy at the same time. Okay, this is a general example of processing in memory and processing in storage and processing in sensors. There's a lot of work going on in this direction that I don't have time to talk about, but if you're interested, you can read some papers that we have been releasing, as you can see, and also some talks that we have been delivering. And the good news today is industry is moving toward this direction. You can see that there's a lot of movement to processing in memory. There are startups that are doing this. There are big companies like Samsung, SK Hynix, Alibaba that have been producing processing in memory chips. And we have been experimenting with UpMem startups processing in memory chips. They have chips that can actually perform general purpose computation inside the DRAM. And these are real. We've been experimenting with them, writing papers. You can take a look at them if you're interested. And we've been developing sequence alignment algorithms that actually outperform all prior algorithms using this sort of system. And more forward-looking, we've also been looking at processing using non-volatile memory technologies, emerging memory technologies. And you can see that there are recent works that tightly integrate different parts of the genome analysis stages in a single chip so that we don't actually move data across many, many systems. Again, I don't have time to talk about a lot of these. This was just a summary. I will briefly mention future opportunities, and future is very bright. I think we have new technologies that are coming up and new applications that are coming up. Some example technologies include nanopore sequencing technology, but there are other technologies, and these introduce new challenges because they're actually very, very high throughput, but unfortunately they have high error rates, as I mentioned, and they have different unique characteristics. Can we design algorithm architecture, our device co-design so that we can actually improve the benefits of these technologies? New applications like graph genomes are important. Reference genomes are getting updated. Can we actually minimize the work that we do if reference genome gets updated? For example, do we actually do the remapping and all of these analyses again and again and again if the reference genome gets updated frequently? We try to minimize that work, and there's a lot of interesting work in this area. So remapping reads between references is actually useful. And there's also other work that you can read on mapping constant regions between references. The key idea is to minimize the useless computation that we do globally in genome analysis. So there's a bright future for intelligent genome analysis in general. Now, let me conclude. Before I conclude, I'll mention that there are things that are happening in the genome analysis industry. Illumina, which is a major technology provider, is actually putting FPGAs in their devices. You can read about this. More recently, NVIDIA has been talking about actually integrating genome analytics. And also, they've introduced general purpose instructions for dynamic programming, which can actually help genome analysis significantly. So where is our dream, basically? We started our dream in 2006 or 2007. We want an embedded device that can perform comprehensive genome analysis in real time within a minute. Unfortunately, we're not there yet, but we made a lot of progress. We need a lot more progress and a lot more effort community-wise so that we can improve energy efficiency, performance, security and privacy, and the huge memory bottleneck that we're facing in this domain. So to conclude, hopefully I've given you a glimpse of the system design for bioinformatics problem, which I believe is critical and has large scientific, medical, societal, and personal implications. I've given you an example of a flavor of this problem, a key step in bioinformatics, which is genome sequence analysis. And we've even focused on read mapping. There are many, many other problems exist, but read mapping is very fundamental because it's the first thing you do when analyzing a genome. We covered some recent ideas. There's a lot more, as I mentioned, but I don't have time to talk about. But hopefully, I've given you a glimpse of many future opportunities that may exist in this domain, especially with new sequencing technologies and with new applications and use cases. So I believe the future is bright, but we also need to do a whole lot of work. And I'd be interested in your thoughts in this problem. Feel free to contact me if you have any thoughts, ideas, or feedback. And there are longer versions of this talk on the internet. This is a version where I delivered a keynote at the Eurosys conference recently in April. But there are other versions also, and I'd be happy to talk in the future. Thank you very much for your attention, and have a great conference. I wish I could be there, but enjoy the conference on my behalf as well. Thank you very much.